The uses of tetramers. A novel and clear technology based on the use of MHC tetramers were developed in the 1990s and made it possible to identify, isolate, and follow the behavior of lymphocytes specific to an antigen of choice. Um, MHC tetramers are laboratory generated complexes of four identical MHC molecules that are bound to four peptides, and they are also linked to a fluorescent molecule. Yeah, a link to a fluorescent molecule. The MHC tetramer staining has been an invaluable tool in our efforts to understand the complexities of lymphocyte behavior in space and in time. You have MHC class 1 tetramers and you have MHC class 2 tetramers. And I have some pictures here. As you can see, this picture here is showing us the binding of an MHC peptide complexes to TCRs and can be stabilized by increasing the avidity through the use of the multivalent MHC peptide complex, which is called a tetramer. So these are tetramers here, and it's undergoing tetramerization. So MHC class 1 tetramers, they were generated first. I guess of course you could think class 1 first. And a given MHC class 1 tetramer peptide complex will bind only those CD8 T cells with TCRs specific for that particular peptide MHC complex. So just like this picture here is described, they said that they basically said the same thing, that MHC peptide complexes binds to TCR. So that's MHC class 1 tetramers basically. This is the process of MHC class 1 tetramers. Then you have MHC class 2 tetramers, and they are powerful tools used to detect antigen specific CD4 T cell responses. They are mainly used to identify and enumerate by flow cytometry and immunohistochemistry. They also do a process of immunophenotyping by flow cytometry. And these two technologies, it made it possible to determine the number and the locale and the phenotype of cells in a population that have TCRs specific for that particular antigen. So just as stated about the MHC class 1 tetramers, they also have to have TCRs specific to a particular antigen. And these two technologies basically made it possible to determine these things of cells, to determine these characteristics and things of cells. And the binding of the TCRs, this to a particular antigen, this can happen before, it can happen after, or it can happen during infection. So the staining, we talked about staining, and it can stain with other cell surface makers to identify various subpopulations. So for an example, like effector and central memory, the staining is a physical approach to the identification of any specific cells, T cells that does not provide functional information. So the staining basically was a way to identify specific T cells, and they can also be combined with intracellular staining of cytokines. For an example, to identify various CD4 subsets would be an example of intracellular staining of cytokines. In addition, tetramers are useful tools to isolate specific populations using a cell sorter. A cell sorter that can purify the fluorochrome, which you can see here in this picture. The fluorochrome is here. So they use a cell sorter to purify the fluorochrome labeled cells within the appropriate gates. Another interesting application of tetramers is that they're used to activate specific ex vivo T cells, and they can colonially expand them because they are composed of MHC molecules and the peptide. They are composed of the MHC molecule and the peptide, which are both essential for antigen recognition and subsequent T cell activation. So, MHC molecule and a peptide, they are two essential things for antigen recognition and for a subsequent T cell activation. So in order for a T cell to activate, it needs an MHC molecule and a peptide.